Hello everyone, welcome back. So today we are going to discuss about genetics. You have already studied almost all of it in your lower classes, but nevertheless, let's start. First of all, obviously the laws of genetics, with the first one being the law of dominance. So obviously you know that we have a pair of chromosomes, and a gene will be present on both and will be represented by a allele, and one of them will always dominate the other one. So for example, if we have a capital A and a small a, and capital A will be the dominant one. So for small a to express itself, it has to be present in a homozygous state because it is recessive one. And if in either case, if it is a capital A and small a and capital A and a capital A, the character which is represented by a capital A will always dominate the other one. Next law, we have law of segregation, that is during gamete formation, alleles for each gene segregate from each other. And so, for example, that capital A, small a, obviously it will be inherited independently into the gametes. Next one, law of independent assortment. The genes of different trait can segregate independently during the formation of gametes. So obviously, as the line itself suggests, that the other genes will always be inherited independently of how other genes are inherited. So let's see a question. This is from the ministry book. That is, a husband is homozygous by a dominant gene, which causes polydactyly, and his wife is homozygous with the recessive genes. For example, we take again, we keep it very basic, and we'll take the letter A. So husband is homozygous with a dominant gene. So if we take the dominant gene as capital A and he is homozygous, and his wife is homozygous recessive, and if we do a cross, so it turns out to be capital A and small a. So which of the below mentioned genetic regularities can be apparent in their children as for having polydactyly? So they are asking that what is this property that all of their children are having polydactyly. So they have already mentioned that polydactyly is caused by a dominant gene, and when we do a cross between this husband and wife, so all of them will inherit a capital A and small a, and capital A is dominant, and this will result in all of them having polydactyly. So this is what Krok mentions that as law of unit characters or law of dominance. So yeah, and these were the other options. I'm not talking about the gamete formation, or they're not talking about two genes. So very easy. Moving on to penetrance and expressivity. So it is mentioned in questions related to this is mentioned in the ministry book. So you should know all these. So penetrance is the frequency of expression in the population when it is present in the genotype. So you can think of penetrance as a binary system. It is either there or it is not there. If it is there, like 1, or if it is not there, like 0. So for example, in this picture, out of all of 9, 5 of them have inherited the violet color, which was supposed to be the dominant one because all of them have, but the others have not. So the gene is penetrant in some 5, while it is not in the other. So when we are talking about the expression of allele in a population, we are talking about the penetrance. It is either there or it is not there. And expressivity is the variation in the allelic expression when the allele is penetrant. So for when we're talking about expressivity, we already know that the gene is penetrant. So for example, hair. So when all of them were inherited with a capital B, which is hair denoted by a violet color. So all of them should be having a violet color, but that is not the case. And so there is some variation seen in all of these. Although the color is present in all of them, that means that the gene for this color is penetrant, but there is some amount of variation, or there's some amount in the concentration of this color. So all of them have different degrees of this color, but nevertheless the color is there. So this is the difference between penetrance and expressivity. Now let's see it with a question. So this is again question from the ministry book, that is question number 69. Inclination to diabetes mellitus is provoked in autosomal recessive gene, and this gene becomes apparent only in 30% of homozygous individuals. So they are saying when it is a recessive gene, and if it is inherited, but not all of them have diabetes mellitus. Only in 30% of the population, diabetes mellitus is seen. So this is the case, and when we look at the options, so obviously you will get confused in either expressiveness or penetrance. So obviously the answer is incomplete penetrance, because first of all they are talking about a population, and second of all they are talking about a binary system. It is either there, that is only in 30% of the population, and in other 70% of population it is not there. So this is incomplete penetrance, and if it was complete penetrance, so anyone who would be inherited by the homozygous recessive gene would be having diabetes mellitus, but only 30% of them are having. So this is incomplete penetrance. Another question, that is, the same genotype in human cell can cause development of feature with different degrees of manifestation. They have already mentioned in the question that there are different degrees of manifestation. And so what do we call this degree of phenotypical manifestation of character controlled by definite gene? And with the same options, so you can see that they are saying that this character, when inherited, has different degrees of manifestation. So it is inherited, but different degrees. So obviously it is gene expressiveness. So moving on to pleiotropy, and people often confuse pleiotropy with polygenic inheritance, polygenic interaction. So, but polygenic interaction is discussed in another topic in later part of this video, but I have also written it here. So, pleiotropy means a single gene produces multiple phenotypic effects. So you just need to remember pleiotropy, one gene, many effects. And polygenic interaction, or how it is mentioned in Krok as polymering, the name itself suggests that polygenic. Many genes control a expression of a trait. So polygeny. And lethal alleles that cause the death of the organism that carry them, like sickle cell anemia. So next, let's also see a question. An 18-year-old male has been diagnosed with Marfan syndrome, and they have written all these various manifestations that are seen with this syndrome. So you know that Marfan syndrome is caused by a mutation in the fibrillin gene. So mutation in one gene, and so many symptoms are produced. So obviously this is what pleiotropy. And for polygenic inheritance, we'll see in the later part of this video. Next is an interaction of allelic genes. So I told you that a gene is represented by two alleles because we have a pair of chromosomes. So what is their interaction? We'll see. So complete dominance. So according to the law of dominance, one allele should always dominate the other, but sometimes that is not the case. But however, if there's complete dominance, what the phenotype of heterozygous will always be the same as phenotype of a homozygous genotype. However, that's not always the case. We often see incomplete dominance. That is, the heterozygous one is almost phenotypically intermediate between a homozygous dominant and a homozygous recessive genotype. And we'll see an example in a question. Co-dominance, that two alleles can be dominant at the same time, and the best example is the AB blood group. So over-dominance means the intermediate has some characteristics or properties that are a little different from the homozygous dominant and homozygous recessive. So for example, in sickle cell anemia, if a person has is heterozygous for this sickle cell anemia, the gene, so you know the disease would not be as severe as it would have been if it was a homozygous recessive, or like a homozygous for sickle cell anemia. So yeah, so first of all, it would not be as severe as like homozygous. Plus, it would have partial resistance to malaria. So this is kind of an over dominance mechanism that is has developed. So let's see a question, that is cystinuria manifests as cystine stones in kidney when it is homozygous, or as an increased level of cystine in heterozygous. And so what is this interaction? So obviously they are saying if it is a homozygous state, there is formation of stone. However, if it is a heterozygous state, there is only increased amount of cystine, but not as much to precipitate a stone. When there's a homozygous like recessive gene, for example, if you take it like so there's no such manifestation seen. So you see that the heterozygous is kind of an intermediate between a homozygous dominant and a homozygous recessive. So it neither has like complete absence of increased cystine, or neither it has that much increased level of cystine to precipitate a stone. So obviously it is what? Incomplete dominance, or how Krok mentioned it as semi-dominance. So let's also see gene interaction of non-allelic genes, that are alleles that are not for the same gene but for different genes. We have complementation. So two genes can somehow complement each other for the manifestation of a trait, and again we will see an example in the next slide. Next is epistasis, that is one gene can mask the phenotype of other gene, and it can be dominant or it can be recessive, and the best example is the Bombay phenotype. So what happens in Bombay phenotype, that H gene, if it is present in its recessive state, it can mask the phenotype of the dominant genes of the ABO system. So here you can see when H was present in a recessive state, it didn't let the B to express itself, so the blood group was O. However, if it was this case like IB and capital H and small h, this gene would not be able to like mask the expression of B blood group. So this would be, the blood group will be B in this case only. Only when the H is present in recessive state, it can mask the expression of ABO system. So next we have polygenic inheritance. Right, a few minutes ago we were discussing about this. So one trait is controlled by several genes, as the name suggests, and the examples can be height, mass, intelligence, skin, eye color, etc. So moving on to question, that they are saying that on X chromosome there's a dominant gene that is responsible for normal blood clotting, and there's another gene that is present on autosomes, and it plays a similar role. However, if any one of these genes is absent, it leads to a coagulation disorder. So obviously, if the two genes are present together, they complement each other and they help in the proper functioning of coagulation. And if any one of these is absent or defective, it leads to a coagulation disorder. So such type of interaction is called complementarity. So next, a woman with O blood group has given a birth to a child with AB blood group, and this woman's husband has A blood group. So you see, how is it possible? Like, how can a child with AB be born when a mother is O? That is, she has no dominant gene of the ABO system, and still a child is having an AB blood group. So in this case, you have to consider the Bombay phenotype. So you see, if we consider the Bombay phenotype, the mother is so the father has A blood group and the child has A B blood group. So it can be like the woman is having IB, but due to presence of recessive H, that is recessive epistasis, this the B expression of B was masked, and hence the blood group was O. Father's blood group was A. But if like father, suppose if the father has a capital H and now the child has inherited capital H and small h from the mother, so it cannot now mask the expression of the dominant genes, and the child now has AB blood group. So there's a possibility if you consider Bombay phenotype. So the answer here is what? Recessive epistasis, because in the woman's case, she was having a homozygous recessive gene, and that was recessive epistasis, and it masked the expression of B blood group, and hence her blood group was O, although she was having the B antigen. So next, this one is from the ministry book, that is intensity of human skin pigmentation is controlled by a few pairs of non-allelic dominant genes, and whenever these genes increase, the pigmentation increase. So you see many genes control one character, that is the skin pigmentation. So many genes means polygenic interaction, or how it is mentioned in Krok, that is polymering. Moving on to variation, a rather easy but topic, and I have like classified it so it is a little easy for you. So variation, it can be heritable or non-heritable. Heritable, since it is heritable, so it must be genotypic, and non-heritable, it is called also called modificative or a phenotypic. And as you can see, combinative mutation can mean genes or in chromosomes, changing the chromosome number or the chromosome structure. So let's first of all look at the questions. So again, question number 92 from the ministry book, that is, parents with normal health have an ill with phenylketonuria son, but owing to a special diet, he has normal development. So what type of variability is his normal development connected with? So first of all, you should know that phenylketonuria is an autosomal recessive disease. So you must so like variation means heritable because it is autosomal recessive and genotype. So it must be like something like mutational or anything other or genotypic. But you see the question in they are asking that what type of variability is this normal development connected with? So owing to a special diet, now he has normal development, but this does not change his genotype. He still has that gene. He still is autosomal recessive for phenylketonuria, and so the phenylketonuria, it is still heritable to his future generations. This normal development is not heritable because it is not changing his genes, so it is not heritable. So here the answer is modificative variability, because if you see in this previous slide, you see non-heritable or modificative. So like similar next question, that a woman got infected with rubella during the pregnancy. Her child was born with malformations, namely cleft lip, cleft palate. His genotype is normal. So like in this question, I've already mentioned that the genotype is normal. So these malformations are manifestation of. So you see, when the woman has something infection or anything that is teratogenic during the pregnancy, the genes were already formed. So his genotype should be normal, although they have also mentioned in the question that the genotype is normal. But like, yeah, so you should read in the question that they have written during pregnancy or before pregnancy. Before pregnancy is also written as a foregoing pregnancy. So foregoing or before. So if it is before pregnancy, there are some chances that the genes are not formed normally. So in that case, it could be heritable variability. But for now, in this question, it is a modificative variability because the genes are normal, but somehow due to environmental factors, she was born with these malformations. But these are not heritable because the genotype is normal. So non-heritable means modificative variability. Next question, similar, that she has taken synthetic hormones during the pregnancy, and her daughter was diagnosed with hirsutism, formerly resembling adrenal syndrome. So such manifestation of variability is called. So again, you see that it is not heritable because the genotype is normal, and all these symptoms are due to the environmental factors, that her mother took some hormones like steroid hormones during her pregnancy, that led to development of such symptoms. But her genotype is normal, so it is this disease or this symptoms, these are not heritable. So when it is not heritable, you should look for modificative variability. And since it is not in the answers, you should choose phenocopy, because it is like a phenocopy. It is phenotypically similar to a disease that is normally inherited, but it is not inherited. It is just phenotypically similar due to some environmental factors. So that was it for the variations. Moving on to transplantation very quickly, 1 minute. Auto means self, so autograft means the graft is taken from the same person. Isograft, iso means similar, so between individuals who are genetically identical. Allo means other, so from non-identical donor of the same species. And xeno means from a donor of completely different species. And with this, we finish our genetics of the medical biology. So obviously, I've not included the chromosomal disorders, etc. I'll probably make a separate video on it. But this was the overall genetics in the medical biology, and I hope you like this video. Please like and share with your friends, and thank you.